great having this meeting here in Mendoza, and also for inviting me to participate in the symposium. What I will do is to talk about acrosomal reaction. And this is a process that occurs with the sperm contact in the proximity of the egg, and is necessary for changing the topology of membranes on the head of the sperm, and that is required for a productive interaction with the oocyte. I choose this micrograph because it was taken by Dr. Burgos in 1955. So in the early days of the electron microscopy, and showed the huge granule that is forming from the Golgi and is near the nucleus. What is secretion? Secretion is the opening of a pore connecting the interior of a granule with the extracellular medium, and that is required for the release of the content of the granule to the extracellular medium. In particular, in the sperm, and this is a human sperm, the granule is weird. You see it's a flat granule going around the nucleus of the sperm. And fusion is also weird because there is not only one single fusion pore, but hundreds of fusion pores. And that will destabilize the membranes in that area of the plasma membrane. And what happened is the formation of these hybrid vesicles. The hybrid vesicles are part of plasma membrane and part of the acrosomal membrane. And at the end of the process, you see that the acrosome is gone, and only this part is remaining, and this part of the plasma membrane of the sperm is the one that is going to interact with the oocyte. That's why you require acrosomal exocytosis for interaction with the egg. Now, how to study the fusion machinery that is here in this tiny cytosol in between the plasma membrane and the outer acrosomal membrane? Because the sperm do not produce proteins, cannot synthesize proteins, and so you cannot overexpress, you cannot knock down proteins in the sperm. Our strategy was to make holes in the plasma membrane using a couple of toxins, streptolysin O or perfluorolysin O. This opened these nice pores. Through these pores, we can introduce recombinant proteins, antibodies, and all kinds of stuff to manipulate the fusion machinery. Another strategy was to use this cell-penetrating peptide, the CPPs. The cell-penetrating peptides are small peptides, about 8-10 amino acids, that when attached to a protein, allow the protein to cross membranes in a way that is not well understood. So we have used this technique to introduce all these proteins. We also are interested in the physicochemical of this process. How come you can go through a membrane? So we made this construct. The CPP attached to a metallothionein, which is a small protein with a high affinity for gold particles. So we have the gold particles, and the gold particles are good because you can follow them by electron microscopy. So when we incubated sperm with this last probe, what we observe is a lot of gold particles inside the sperm. At the higher magnification, you see the spot, the black spot are the gold particles and are crossing the plasma membrane. One good thing with the acrosome, it is large. So you can see it in the light microscope. In this case, it's a fluorescent microscope. On controlled conditions, the green stuff, like a hat, is in most of the sperm. The nucleus is in red. And upon stimulation of secretion, many of the sperm lost the acrosome, and you see there is not a green hat. So with these very simple techniques, we have a model, a working model for secretion. We think that an initial entrance of calcium from the extracellular medium is required to activate the fusion machinery. The fusion machinery are, for example, these snare proteins that require to be reassembled in trans, bridging the two membranes that are going to fuse. And then the fusion pore can open and secretion occurs. This was important. Our observation indicate that at this point, you require an entrance of calcium from the acrosome. That's what I'm showing here. This entrance of calcium coming from the acrosome is required for the opening of fusion pore. In other words, we're thinking that you need calcium to get in to activate all the fusion machinery, and you need calcium to get out of the acrosome to open the fusion pores. We arrange the cascade of events that occur in this process in two branches. One is the one that is going to activate snare proteins, and the other is the one that will activate the release of calcium from inside the acrosome. So we have a very strong prediction here. We are thinking that this part of the pathway where calcium activates cyclic AMP production and that activates this protein called EPAC is activating the release of calcium from the acrosome. Can we test this directly? And this was done by the group of Claudia Thomas, and what they did was permeabilize the sperm, load the acrosome with a calcium-sensitive fluorescent probe, so she can follow the concentration of calcium in the acrosome. And then when they add calcium here in the cytosol, what you see is a calcium-induced calcium release. The calcium is gone from the acrosome. According to the model, this should be sensitive or should require cyclic AMP. So she showed, and the group showed, that if you block the production of cyclic AMP or you deplete from cyclic AMP, you block the effect of calcium. And if you activate with this cyclic AMP analog, which is specific for EPAC, you observe the release of calcium. But if you use an antibody that block the EPAC, it will block the release of calcium from inside the acrosome. So this is the beautiful work done by Ornella Lucchesi in the group of Claudia Thomas. Another thing that we observe when we block the release of calcium from inside the acrosome is that, of course, the exocytosis was blocked. But when we look at the electron microscope, the sperm, we observe that the acrosome has a very weird shape. It was swollen. There was a lot of invaginations of the outer acrosomal membrane. There was the formation of internal vesicles. And we think that this swelling is part of the fusion of the secretion machinery. We speculate that the swelling allowed the contact between the outer acrosomal membrane and the plasma membrane, and this invagination will shape a future hybrid vesicle. If you have fusion here and here, you will release this as a hybrid vesicle. So my group now is trying to understand the mechanism of swelling. We know that swelling does not require the release of calcium from inside the acrosome because we can observe it when we block this. So it should be something over here. It could be calcium, it could be cyclic AMP, and, or EPAC. What we did was try to see if we can induce swelling in the absence of calcium by adding cyclic AMP, and that's what we observed. Even if we had BAPTA to chelate calcium outside, BAPTA AM to block calcium inside, we can get swelling with cyclic AMP or this analog that activate EPAC. This was in permeabilized cells. In non-permeabilized cells, if we induce with progesterone swelling, we observe that we block swelling with some drug that inhibit the production of cyclic AMP, or if we use this sponge has a CPP, That's why we can use it in non-permeabilized sperm. So we think that swelling is triggered by increase of cyclic AMP. One observation in our model is that this initial calcium entrance is not immediately cause exocytosis. This initial calcium entrance, and I'm just with some difficulties because in this the mouse doesn't work very well. But okay. What we observe is that here. No. Okay. What we observe is that when we induce exocytosis, calcium gets up very quickly. You see the blue? In seconds, you have calcium inside the cell. But exocytosis that is measured in green is occurring in the next 10, 20 minutes. We need about 14 minutes to get half of the sperm to react. So what is the kinetic of swelling? It's our question. What we observe is when we measure swelling across time, we need 13 minutes for half of the sperm to have a swollen acrosome. So it's a slow kinetics. And one question is, when the acrosome is swollen, how much will take for the final opening of the fusion pores? And we could do that because this inhibitor is reversible. So we can illuminate, and that is light sensitive. We destroy the inhibitor, and we can measure what is the kinetics of the last part. And the kinetic of the last part is very fast, about a few seconds. So our conclusion is that the swelling has a slow kinetic that is consistent with the slow kinetics of exocytosis. You remember that we said that we believe that this apposition is part of the fusion machinery. And if you remember the scheme, the previous one, snare should be there, should be bridging the two membranes that are going to fuse. So what happened when we destroy snares? When we hydrolyze snares using neurotoxins like a tetanus toxin or botulinal toxins? Well, this tight apposition, this is measuring the distance between the two membranes. The tight apposition, less than 8 nanometers, that is very frequent in normal condition, decreased significantly when we destroy the snares. And you remember that we have all these internal vesicles and all these invaginations, and this is very similar what happened with multivesicular endosomes, that you have the invagination of the membrane of the endosome to form vesicles. And that is require a series of complexes called escort complexes. So what we did was to interfere with the function of escort complexes using 4 different proteins and 4 different antibodies, and all of them block secretion. This was done in permeabilized sperm. This was done in non-permeabilized sperm using one of these proteins attached to a CPP. So now we know that snare and escort are part of the mechanism of the function of the swelling. If you observe a swollen acrosome, you see the huge increase in volume. If there is a huge increase in volume, you would think that water is coming in. And water to go through a membrane requires some proteins that are called aquaporins. So we try to inhibit aquaporins and to see what happens with swelling, and we use a couple of heavy metal salts, mercury and silver, and they block swelling, and also an antibody against aquaporin. We think then that aquaporin are part of the mechanism of swelling. We have some progress, but it's not so much progress. We're going very slowly. And one of the reasons is because the only way we have to assess swelling is by electron microscopy. An electron microscopy takes a long time. So we're trying to see one other way to detect swelling. And this is the idea of Gerardo de Blas. Okay, he thought, well, if we load some fluorescent probe inside the acrosome and we induce swelling, that fluorescent probe will be diluted, and we will see a decrease of signal upon swelling. And this is what is not working? Yeah. Okay, no matter. The video shows that this label here goes down when we induce swelling. No? This is the measurement of that decrease of signal. And is this swelling or not, or something else? We can block by using the heavy metal salts, silver or mercury. So it's a good chance that we will do much more progress in the next future using this technique. And I will acknowledge the people working. The first observation was done by Natalia Zanetti, working in the swelling, and she characterized the effect of neurotoxins. Christian Pocognoni was working with the ESCOR. Victoria Berberian is right now actively working with CPPs and this project and several other projects. Claudia Sosa worked with the cyclic AMP and swelling. And this is the group of Gerardo de Blas. Esperanza Mata is coming from Darson's lab, working with Gerardo in the characterization of this real-time assay for swelling. And Rodolfo Arias is part of the group. And we have other groups with the other projects here. And thank you very much for your attention.